Hello everyone. Thank you for joining us. Welcome to the Avexia webinar series. Our topic for today is genetic predisposition, genes that could influence cortisol, neuroplasticity, and focus. My name is Christopher Chu. I am the Director of Marketing for Avexia Diagnostics and will be your host for this webinar. Today I have the distinct pleasure of being joined by an exciting guest, Dr. Russell Amato and Dr. Jordan Plummer, who will be our presenters for this webinar. Dr. Amato is currently a Medical Affairs Director with Genemind. Dr. Amato received his PhD from the Department of Pharmacology and Experimental Therapeutics at Louisiana State University Health Science Center. Dr. Plummer is currently a Medical Science Liaison in the Midwest region. She received her PharmD from Temple University, where she had an internship with Genemind and graduated from Temple as a class valedictorian. Without further delay, I will now turn the webinar over to the speakers.
Good afternoon. I'm Dr. Russell Amato, and I'm going to be starting our talk off on genetic predispositions, genes that can influence cortisol, neuroplasticity, and focus. And we're doing this in partner with Avexia Diagnostics. Now, to introduce you to the speakers for today, as I mentioned before, I'm Dr. Amato. I'm currently the Medical Affairs Director for Genemind. I graduated with my PhD from Louisiana State University Health Sciences Center. Spent the first 10 years of my career in academia publishing on novel therapeutics and preclinical models of psychiatric disease. And that included things like depression, anxiety, drug dependence, cognitive dysfunction. I started working for Genemind about 9 years ago because I wanted a more direct impact on patient lives, not so much publishing a bunch of papers that only a very few select academic scientists will ever end up reading. And with me today, I also have Dr. Jordan Plummer, right? And she received her PharmD from Temple University. She was at the top of her class there. Dr. Plummer actually did an internship with Genemind while attending Temple University, and we were so impressed with her work ethic and ability, we offered her a medical science liaison role here after she completed her postdoctoral work at Merck, where she kind of gained a lot of expertise in various areas of medical affairs across the variety of different therapeutic areas. For today, we're going to be looking at, well, three parts of our talk, right? An evolving perspective on mental health, genetic predispositions, and then we're going to actually dig into the individual genes themselves. But just to give everybody kind of a deeper dive on this particular slide, basically the genetic predisposition portion is going to be about getting to know the genes that might influence your cortisol release, right? It might influence your neuroplasticity. It might influence your cognitive, right, abilities. And while it's well established that our behaviors are influenced by our environment, experience, and genetics, I do like to make sure that most people are aware that genes generally tend to have a small influence on behaviors. Some can create a predisposition towards certain characteristics or traits. Today, we're going to discuss genes like BDNF, COMT, SLC6A4, and the mechanisms by which these gene variants can create a bias towards some behaviors. We'll also discuss supplements that may target the biochemistry and the physiological changes associated with these genetic variants. And so let's go ahead and dive into it. All right. Excellent. So first, we're going to go through the genetics. You may recognize this as the old Mendelian genetics and Punnett squares. Remember high school? The big take home for this slide is that we can inherit different variants of the same gene from each parent. These different variants may end up having different biological consequences depending on which one we received and whether it's dominant or recessive, right? As well as how many alleles we carry total. Now, when identifying gene variants that might impact our traits or behaviors, we originally started by identifying candidate genes, right? This was 30, 40 years ago. We were looking at pre-specified association studies. So one example of by what I mean by pre-specified is that, well, we could be looking for genetic variants of the dopamine receptor that might predispose somebody to developing a drug dependence because of all the research that we had previously done on the dopamine reward pathway, right? We were looking for a specific association with that behavior in a pathway that was pretty well studied, right? Pretty well replicated. However, a more common, newer way of identifying these genes is to look at one group of people versus another group of people called genome-wide association studies, or GWAS. And these people, like the comparison you're making is against, well, let's say you have 1,000 bipolar patients, right, or patients with bipolar disorder, and you compare them to 1,000 non-bipolar control subjects, right? Maybe they have no mental health issues, they're just control subjects. Well, what those analyses do is they look through all of the genes, looking for any genes that are more common, prevalent in the bipolar group compared to that non-bipolar group, right? So again, we don't know which gene specifically we're going to run into, which variant we're going to find. It's just basically analyzing every single gene variant and finding the ones that are most commonly found in that particular population. Now, this has led to kind of the newest area of research, the one that we have, unfortunately, the least amount of data on, and they're called polygenic risk scores, or you can see it up on the screen here. It says PRS. These are focused on the additive or combinatorial effects of multiple gene variants and their associations with specific behaviors, traits, and drug response. And these types of studies give us a more comprehensive view of our genetic predispositions and may one day even help aiding in diagnoses. To give an example of each of these types of research, let's walk through the process to get a more total or comprehensive understanding, right? One example of the candidate gene approach focused on researchers hypothesizing that because major depressive disorder can be treated by SSRIs, that there may be genetic associations with the biological target of this drug class. More specifically, they investigated gene variants in the serotonin transporter that might impact responses to these SSRIs, as well as changes in mood-related behaviors. And we're going to talk about some of those studies later on today when we go to the individual genes. For the genome-wide association studies we previously mentioned, that specific genes were isolated as potential predispositions for bipolar disorder, well, two genes came out of that study, right? The ANC3 and the CACNA1C, which regulate the flux of positively charged ions across the neuronal membrane. And those data were actually replicated across multiple genome-wide association studies. But the increase in absolute risk was pretty small. News headlines, which seized on the more relative risk increases, touted that these genes could increase your risk for bipolar by 16 to 38%, which sounds like a pretty large effect. However, any good scientist will be aware that those news organizations like to use relative risk, so the impact appears larger and is more newsworthy. When looking at the absolute change in risk, you'll actually see the increase is small. As an example, if your initial risk for developing bipolar disorder in the general population is 1%, well, by inheriting these risk variants from the CACNA1C gene or this ANC3 gene, well, your new risk may be closer to 1.16 to 1.38%. That doesn't sound nearly as alarming as a 16 to 38% increase in risk, and that's because we're talking absolute risk now. So this is actually one of the reasons why scientists wanted to look at polygenic risk scores, because, well, we might need multiple genes in order to be able to assess more of that risk. Specifically, these studies looked at multiple genes, hundreds in the case of the latest polygenic risk score for bipolar disorder, and they tried to place these individuals into buckets of risk. Well, what do you do with that info? Well, you use the score plus any environmental information you have, like family history, to place people into different buckets of intervention, right? If somebody has a high PRS risk score, do we give them earlier intervention? Do we get more aggressive as far as treatment goes? While these techniques are improving how much absolute risk we can account for, unfortunately, polygenic risk scores, PRS studies are still kind of in their infancy, and there's still a large environmental component when it comes to risk. So I don't think we're quite there using it clinically yet. One of the ways, however, we might start using these polygenic risk scores is to basically gauge that patient's total risk level, look at their total level of environmental stress. And if your patient has a low risk PRS score and is doing everything right environmentally, you might choose to treat them with a pretty standard intervention, right? If those same people with a low PRS score were eating poorly, smoking, experiencing significant trauma, we might actually want to go into that early intervention despite their low PRS score, right? Just the environment alone may be enough to, right, intervene sooner. However, if the patient has both a high risk PRS score and poor environmental conditions or modifiers, those candidates may be candidates for both early intervention and more aggressive treatment strategies, right? So we really want to get in there because they're already genetically predisposed to having some of those symptoms and behaviors. Now they've got this environmental trauma stacked on top. And so we want to make sure and have the most aggressive treatment for these types of patients. Now, what is a predisposition? Well, a genetic predisposition refers to a genetic result that can increase the likelihood of having a certain behavior or trait that can impact health and wellness. Patients may wonder about the implication of their genetic results, but it's important to remember that our genes are not our destiny, right? Inheriting some of these risk variants from mom and dad may not necessarily make us depressed, make us anxious, make us bipolar, what have you, right? In several cases, a genetic predisposition towards a specific biological mechanism can be just as important as a learned behavior or a trait which an individual possesses. However, I do always end on the caveat that a single genetic variant overall tends to have smaller overall influence, right, on behaviors. But knowing the patient has that predisposition puts us in a position to do something about it early. The benefits of knowing a patient's genetic predisposition is that we can keep an eye out for certain traits they are more likely, on average, to express. And that may help us make an earlier intervention, a stronger intervention. However, before we proceed with that, we really do need to validate that with patient presentation, right? We need to do our appropriate clinical history. We need to bring in those tools like the neuropsych evaluation, right? We're still doing good medicine. We're just adding another layer. Just because somebody has a genetic predisposition does not mean that they will have that trait necessarily. Now, for today's presentation, we'll largely be focusing on 3 genes involved with stress response and resilience. We'll be looking at the serotonin receptor, also known as SLC6A4, brain-derived neurotrophic factor, also known as BDNF, and the catechol-O-methyltransferase, also known as COMT. Variants in these genes can lead to changes in expression and structure of the encoded protein, with downstream physiological changes that may alter the way in which we respond to environmentally induced stress. And we're going to start with this gene, SLC6A4, the serotonin transporter. So on a typical pharmacogenetic report, we look at variants in the serotonin transporter as a predictor of SSRI response and risk for adverse events. This may be due to changes in how much of the protein is expressed. More specifically, patients that inherit a variant within this gene can actually decrease the number of serotonin transporters on the cell membrane. That can lead to changes in how much serotonin is available in the synapse, for instance. There are 2 variants of this gene. They're called the long and short. We call it short because this version of the gene experienced a 44 nucleotide deletion at some point in the distant past, right, as we were evolving. And if you inherit two copies of the long gene, you produce the largest amount of serotonin transporters, and these individuals can efficiently clear the synapse of serotonin so the postsynaptic neuron isn't overstimulated. These individuals tend to respond better to SSRIs, at least based on the data that we have, and tend to have fewer side effects from this medication class. However, in people that carry a short gene, this can lead to underproduction of these serotonin transporters and an inability to efficiently remove serotonin from the synapse. As you can see in the animation on the slide, right? It's like a ton of serotonin in there. Scientists hypothesize that when we give an SSRI medication, which may further reduce the activity of these transporters, the patients may become overexposed to the serotonin, and this may be why we see increases in adverse events and even medication failure when patients carry the short version of the gene. Newer research has also identified two versions of the long gene that may also have different activity. An LA variant, where an adenine has been replaced with a guanine, that we designate that LG, well, it has similar activity to the short variant. It is associated with reduced serotonin receptor expression. This slide does a nice job of bucketing the SSRI response based on your genotype, right? But the section we are most concerned with, right, with today's talk is how it may impact changes in physiology. So in patients that carry the short variant, these patients may be more predisposed to having a pronounced cortisol response to stressful life situations, right? Stressful environmental events. This study was done by Ian Gottlieb at Stanford in 2008. They asked 9 to 14-year-old girls to count backwards from 400 by sevens as quickly and accurately as possible. If the participant made a mistake, the experimenter stopped them. They were told to start over again. Now, that was a little stressful, being told you were wrong, but not too stressful. And as you can see on the graph presented, participants with two versions of the short serotonin transporter were much more reactive to this task compared to the other subjects. And they did not go back to baseline for 45 minutes to an hour longer, right? So they had this more prolonged stress response, more prolonged fight or flight. Now, several other studies replicated these findings, including the study you're looking at right now from Baldwin Way at UCLA, in which they took 182 young adults, put them through the Trier social stress task, which is, getting them up in front of their peers and making them do a bit of public speaking, which can be pretty stressful for most people. In the graph presented, you can see that when there was no audience to speak to, all three gene profiles didn't really see any significant change in stress levels, right? Cortisol release. However, when placed in front of an audience, that's the graph on the right, well, subjects that carried the two short versions of the gene experienced a stronger, more significant increase in that cortisol release, right? Stronger stress response, which you can see there in the blue line in that graph to the right. But rather than spend all of today looking at the serotonin transporter, I'll actually turn the talk over to Dr. Plummer at this point so she can give a more in-depth dive on some of the other genes like BDNF and COMT in a little bit more detail and about how they affect things like stress response.
Awesome. Thank you, Dr. Amato. So let's move on to BDNF. If you joined us for the last Genomind and Avexia webinar, you might remember this gene. We highlighted its important connection to exercise, which we'll touch on again briefly today, plus a couple other studies that we'll discuss as well. So as a quick refresher, think of BDNF as a key marker of neuroplasticity, right? It literally helps increase the connections between neurons. And BDNF is essential for healthy neuronal signaling and helps the brain stay resilient in the face of stress or damage. And there's several ways we can boost BDNF production, right? Exercise is one of the most effective, especially weight-bearing activities, since our skeletal muscles actually release BDNF, and some gene types respond particularly well to this. Enriching environments and even some antidepressants can also help increase BDNF levels. It's also important to know that there are two common variants of the BDNF gene, VAL and MET. Most people have the VAL-VAL type, which produces normal BDNF levels, but individuals with one or two MET alleles, so VAL-MET or MET-MET, typically secrete less BDNF and tend to show things like more PTSD symptoms and working memory difficulties, which has been shown in various different papers and research, right? So overall, we want to focus on ways to increase BDNF to support brain health and resilience. As we covered in the last Avexia webinar, so I won't go into too much detail here, but here's a quick recap on BDNF and exercise, right? So exercise boosts BDNF, which plays a key role in cognitive function. On the left, we see working memory performance by genotype. Our VALs are our solid lines, and MET carriers are the dotted line. MET carriers start lower, but improve rapidly with exercise, eventually surpassing their VAL individuals or counterparts. This suggests exercise can help overcome that genetic disadvantage. Now, on the right-hand side, PTSD symptom severity in veterans shows a similar pattern, right? MET carriers start with more severe symptoms, but experience significant improvement with exercise, unlike their VAL individuals. So again, physical activity appears to offset that genetic vulnerability. This, however, is something we didn't cover in the last webinar. What's fascinating is how the MET allele can affect anticipatory stress. So in one study here on the slide, participants were told they would undergo a stress test, specifically placing their hand in cold water for a few minutes. Before the test even began, individuals with the MET allele showed the highest level of anticipatory cortisol release, meaning their stress response was triggered just by the expectation of the upcoming challenge. Interestingly, once the participants actually put their hand in the cold water, cortisol levels were the same across all genotypes. So what this tells us is that MET carriers may have a more pronounced anticipatory stress response. Their cortisol levels may rise more in response to the anticipation of stress itself rather than the actual physical experience. Interestingly, restoring BDNF levels may play an important role in how certain medications work. What we have on the slide here is a study by Rickett and colleagues that looked at this connection by examining BDNF levels during lithium augmentation. They studied 83 patients who hadn't responded well to antidepressant therapy, and BDNF serum levels were measured before and after 4 weeks of lithium augmentation. The results showed a significant increase in BDNF levels during lithium treatment, and important to mention that this was seen with therapeutic doses of lithium. Interestingly, though, other research has shown that even small amounts of lithium, such as in supplement form, may also help increase BDNF levels too. From a diet perspective, there are several things you can do to potentially help your gut bacteria produce short-chain fatty acids, which are also known to boost BDNF levels. One of the most effective strategies is increasing soluble fiber in your diet. This feeds the gut microbiome and stimulates the bacteria to produce these short-chain fatty acids, supporting brain health. So butyric acid, for example, a short-chain fatty acid, has been shown to potentially increase BDNF levels as well. So the key takeaway here is that if you carry the MET allele, there are a variety of lifestyle strategies you can use to potentially help offset its effects and increase BDNF production. Remember, though, like we mentioned, your genes are not your destiny. The last gene we'll cover today is COMT. We'll be looking at how it impacts the frontal cortex, specifically its role in regulating dopamine levels in the front of the brain. So let's talk about why this is important. There's something called the dopamine U-shaped curve. You see this here on the slide. Also like to call this the Goldilocks principle for dopamine, right? We know too little dopamine isn't good and too much dopamine isn't good either. And looking at the image on the screen, as dopamine levels increase along the x-axis, we'll see improvements in working memory. This peaks, right, at an optimal point, and then if dopamine levels continue to rise, working memory starts to decline. So it's about finding that optimal balance or that balance between the two. Now, let's talk about genetic differences. There are three versions of the COMT gene: VAL-VAL, VAL-MET, and MET-MET. Our VAL-VAL individuals metabolize dopamine faster than others, leading to lower dopamine levels in the frontal cortex. They start on the left side of the curve and would potentially benefit from increasing dopamine to reach that optimal range. Our VAL-MET individuals typically start in the optimal range. Most of us fall into this category. And then our MET-MET individuals on the right do not break down dopamine as effectively, resulting in higher dopamine levels at baseline. They start on the higher end of that curve. So increasing dopamine in these individuals may worsen their working memory. So this explains why there's been some clinical research here that has shown that second generation antipsychotics may be more effective for our MET-MET individuals, while some of our stimulants like Adderall or Ritalin may be more effective for our VAL-VAL individuals. But how can this affect our behaviors? Historically, the COMT gene has been described using the term warriors and worriers, right? Our VAL-VAL individuals have been known as the warriors. They tend to have lower dopamine levels in the frontal cortex, making them more impulsive and better at handling stressful situations. Our MET-MET individuals have been known as the worriers. They have higher dopamine levels, leading to an anxious phenotype, some cognitive rigidity. Stressful situations may decrease their cognitive function and accuracy. So, for the figure on the left here on the screen, in a working memory test, those shaded squares represent the vowels. And if someone's stressed out a VAL-VAL, their working memory actually improved. However, if a MET-MET was stressed out, they shrunk under the pressure and their cognitive function decreased, showing that phenomenon of VAL-VAL individuals may be thriving under pressure while MET-MET individuals struggle a little bit. The same outcome was shown for the figure on the right there. So, as a takeaway or in summary, what we've seen with some of our COMT VAL-VAL patients who metabolize dopamine faster, where they have lower levels of dopamine in the prefrontal cortex, maybe tend to have poorer memory, poorer attention control, more resilience to stress, and a higher pain threshold. Our COMT MET-MET patients who metabolize that dopamine slower, they have higher levels, tend to have better memory, better attention control, but less resilience to stress, and a lower pain threshold. Studies have shown that MET-MET individuals may perform better on cognitive tasks, but are at risk of that cognitive perturbation, which includes repetitive obsessive behaviors or thoughts, and that elevated dopamine levels in the forebrain are believed to cause these behaviors. So that's just in summary what some of the research has shown on COMT, but I can't stress this enough, right? That our genes are not our destiny. If you have these gene types, this does not mean these behaviors are something you will experience, maybe ever. So just wanted to highlight that as well. The last piece here I wanted to cover are some supplements that we may be able to use, right? For individuals with the VAL-VAL genotype, we could consider, because their COMT is hyperactive, we could consider natural COMT inhibitors to balance out these dopamine levels. So EGCG, which is found in green tea extract, and quercetin may be helpful in boosting the dopamine naturally. And then on the other hand, for our MET individuals who have those high dopamine levels because their COMT is hypoactive, we could potentially utilize supplements that help decrease cortisol, like our cortisol adaptogens such as omega-3s, ashwagandha, and magnesium, just to name a couple that may be beneficial in these situations. And lastly, just wanted to show a study in a summary slide to kind of encapsulate all the genes we've discussed today, right? Three of the most studied genes in the HPA axis dysfunction and fear potentiated startle are actually COMT, SLC6A4, and BDNF. In all three of these graphs, you can see that certain genotypes, specifically our COMT MET METs, our SLC6A4 shorts, and our BDNF MET allele carriers, tend to release more cortisol in response to social and physical stressful tasks. So I think this slide does a really good job of kind of taking all three genes Dr. Amato and I talked about today and giving an example of how certain gene types tend to lead in one position over another when it comes to that cortisol release. And with that, I think we've covered the key insights for today's session. We hope you found this information helpful and gained some fascinating insights into how our genes could potentially predispose us to certain behaviors. So thank you all for joining us.
For any questions or issues you may have, contact Avexia Diagnostics by email at info@avexiadiagnostics.com or by phone 5 days a week at extended hours 8 a.m. to 8 p.m. Eastern Time at 888-852-2723. Thank you, Dr. Amato and Dr. Plummer, for this informative presentation. Thank you all again for joining us for this webinar event. Until next time, from everyone at Avexia Diagnostics and Genomind, stay healthy, stay safe, and we wish you all the best on your pathway to wellness.